Fiber optics is already revolutionizing communication networks, from telephone systems to the internet to media displays. But could it also someday help battle neurological diseases such as Alzheimer's? Hello, I'm Justine Murphy, and this is Light Matters for April 22nd, 2016. Thanks for joining us. Researchers at MIT are experimenting with a new optogenetics technique that uses fiber optic light stimulation to ultimately regrow lost synaptic connections in the brain. They are currently experimenting with a mouse model of Alzheimer's disease and have already made some incredible discoveries. But we'll get to that a little later. Also on tap for this month's show is an IR imaging method that is helping beekeepers ensure the health of their colonies. And managing editor Michael Wheeler is here to talk about how growing nanostructures could someday create self-cleaning textiles. But first, there's a new non-invasive spectroscopy technique for the assessment of bacteria in food, blood supplies, and more. Called TUNABLE diode laser absorption spectroscopy, this new technique allows quick, accurate, non-invasive measurement of bacteria levels in products that are derived from living matter. It is the most common laser-based absorption technique for quantitative assessments of species within a gas phase, according to a team from the Institute of Information Optics at Zhejiang Normal University in China. And it can be used to measure the concentration of specific gaseous species, such as carbon dioxide within gaseous mixtures, through absorption spectrometry based on tunable diode lasers. So, since the production of carbon dioxide is associated with the growth of microorganisms, the researchers found that the technique could also be used to measure microbial growth by evaluating the level of carbon dioxide within a closed compartment, such as a bottle or a bag. To do this work, the Zhejiang researchers collaborated with a team from Umeå University in Sweden to develop an instrument based on the laser absorption technique. It allowed them to assess bacterial growth of various types of samples under a variety of conditions. The device's basic setup involved a tunable diode laser as the light source, beam shaping optics, a sample to be investigated, receiving optics, and one or more detectors. In the future, the researchers plan to enhance the tunable diode laser absorption spectroscopy technique to allow the measurement of microbial growth in samples beyond food items and medical supplies.
Imagine hanging your clothes on the line for laundering instead of drying. As futuristic as it sounds, that may one day be a reality thanks to recent findings from researchers at RMIT University in Melbourne, Australia. The work could lead to mass-produced, nano-enhanced textiles that can clean themselves when worn in the sun or exposed to light. Led by researcher Rajesh Ramanathan, the team developed a cheap and efficient way to grow nanostructures based on copper and silver. The nanostructures receive an energy boost that creates hot electrons, which release a burst of energy that degrades organic matter. Growing nanostructures with these capabilities had been difficult outside of the lab. Using a fairly simple technique that involved dipping the textiles into different solutions, they were able to develop stable nanostructures within 30 minutes. The first commercial products incorporating the nanostructures may come within 3 to 5 years, according to Ramanathan. Until then, hold on to your whirlpool.
A new experimental optogenetics technique is providing a better understanding of memory retrieval, which could potentially help alleviate symptoms of Alzheimer's disease. Developed by researchers from MIT's Riken Center for Neural Circuit Genetics, the technique was applied to a mouse model of Alzheimer's disease. They found that spines, which are small knobs on brain cell dendrites that facilitate the formation of synaptic connections, are essential for memory retrieval. In addition, fiber optic light stimulation could help regrow lost spines. In the study, the Alzheimer's mice received a foot shock while inside a particular cage. Remembering and expecting those shocks caused them to freeze in that particular enclosure, but not in a neutral one. According to the researchers, compared with normal mice, these experimental rodents actually exhibited amnesia and reduced freezing behavior, which indicated progressive memory loss. Memory traces, or engrams, of the foot shock experience were found in the dentate gyrus of the hippocampus, which is a key area in the brain for memory processing. A virus was used to deliver a gene into the dentate gyrus. It labeled active engram cells and allowed visual identification of the memory trace neurons for that specific foot shock memory. A second virus was then used, which contained a gene that made these engram neurons light sensitive. When reactivated with light in the Alzheimer's mice, memory of the foot shock experience became retrievable and their previous behavior was restored. Through repeated stimulation with high-frequency bursts of light to the hippocampal memory circuit, the number of spines increased. These results suggest that impaired retrieval of memories may trigger what is a prominent symptom of early Alzheimer's disease, and the synaptic connectivity between memory cells could be crucial for memory retrieval. There's a buzz around IR cameras, and it's getting louder. Last January, the Utah Department of Agriculture and Food, UDAF, launched one of the first winter honeybee hive inspection programs in the U.S. It uses a forward-looking infrared camera to detect colonies that have been decimated by mites or disease. This has shown beekeepers whether a colony is emitting a strong or weak heat signature, enabling them to accurately gauge the health of a colony without having to open the container in which it is housed. The IR camera used in the study could replace beekeepers' traditional method of hive inspection, which has been to place a stethoscope on a hive container to listen for the buzz of bees. However, with that traditional method, it's tough to determine just how many bees are buzzing, according to UDAF. With the IR camera, the heat signature it captures provides a clearer picture of the hive's population. This helps beekeepers segregate those colonies infected with diseases such as American or European foul brood and dispose of contaminated equipment used in hive containers. This work is necessary to preserve the vital role honeybees play in Utah's economy, according to the researchers. IR cameras serve as a tool for beekeepers to ensure that preservation.
On the first Saturday in May, many of you will watch the Kentucky Derby. There are few sporting events that offer such a visually rich experience for the TV viewer at home. In the May issue of Photonic Spectra, we look at one of the underlying technologies, quantum dots, advancing consumer displays to enhance that viewing experience. The article examines a heavy metal-free version of quantum dots that are capable of delivering the same color purity and brilliance as their predecessors. Also, we examine the singular role of the photon for ensuring data security over today's expanding quantum communication networks. And we delve into the shifting landscape of laser machining, where fiber lasers are increasingly being used for cutting, welding, ablation, and marking. You can read all of this and more in the May issue.
Thanks, Mike. Photonics Media offers a full lineup of free webinars presented by industry experts from around the world. We've got quite a few coming up soon, focusing on topics like bioluminescence imaging, OLED displays, and tips for commercializing photonics technology. Past webinars are available on demand too. To learn more and to register for upcoming presentations, visit photonics.com/webinars. And that's it for this month's show. If you haven't already, check out the newly redesigned photonics.com. And be sure to follow Photonics Media on LinkedIn, Facebook, Twitter, and YouTube. Thanks for watching. We'll see you next time.